Hi, Dr. Tom here. Hey, and I just want to highlight a couple of things about the Desert Microbe STEM Challenge that I think will be helpful for you. So first of all, guys, the main thing that you use in the Microbe STEM Challenge is you use water lock powder or water gel powder. You can get it from Steve Spangler. The chemical name for it is sodium polyacrylate. There is a container of it, and that's what you use as the main substance for your desert microbe. So what you do then is take about 20 or 30 grams of the desert microbe, the white powder, and put it onto a paper plate or something like that. And then to make it more microbe looking, you want to put on your safety goggles, and also make sure your kids wear safety goggles in this experiment. And then you add some drops of food coloring to the white powder, and then you kind of mix it in using a spoon of some sort, and this takes a little bit of time, but you mix it in like that, and eventually the powder will become green and much more microbe looking and much more realistic looking overall. All right, once you do that, then what you want to do is take a small portion of this and divide it up for each group of kids. We try to take about 0.5 grams of microbe for each group, and then we put it in a little plastic container like this, put a little M on the top to make it look a little bit more authentic. All right, and then kids take it out and we have them put it on just a little styrofoam plate, and then the STEM challenge becomes this: how much water can this desert microbe actually absorb? And along the way we talk about the fact that, you know, desert environments are very dry, it doesn't rain very often, so they have to be able to absorb a lot of water and then conserve a lot of water too if they're going to be able to survive. So we kind of bring this out. Here's this newly discovered desert microbe. Our challenge is to figure out how much water can it absorb before it's saturated. And so to start with, the kids actually take a little pipette and start adding the water just a drop at a time. And we tell them that about 20 drops equals 1 milliliter. So they're calculating how much water they add. And right from the get-go, it's amazing how much water the microbe can actually absorb and how well it absorbs the water. And so kids might do this for 40 or 60 or 80 drops, but pretty soon they start getting a little bit tired of it. And so we say, hey, if you want to use a graduated cylinder and just use measured amounts of water, you can do that too. It might go a little bit faster. And so kids will get like a little graduated cylinder like this. This one has 35 milliliters in it right now, and they can start adding it by just pouring it on there kind of slowly and seeing how the microbe continues to absorb water, and then making sure that they measure how much water that they've added each time. So in this case, we've added about 11 milliliters of water, but you can see the microbe seems like it can absorb more water yet, and so they keep adding and keep doing this as they go. All right, and as it turns out, the microbe can absorb a tremendous amount of water. And so this might be about 1/5 of the saturation point right now. If we look, this one right here, this might be about 1/3 of the way to the saturation point or so. So we just keep encouraging our groups to keep adding it and keep checking it out. And they really have to decide, what do they think it means to be saturated? All right, but eventually then students keep adding, and this one is a microbe that's close to the saturation point. Might be able to take a little bit more, but you have to just keep adding and adding until you think your microbe is saturated. And then once that's done, guys, we have the groups go and put their data on the board. We kind of compare the data, and we have a fair amount of variability from group to group, but that's kind of should be expected, you know, keeping in mind that everybody's kind of coming up with their own definition of saturation. And some of our groups are being very careful and very systematic. And as you know, some of our groups will be a little bit more haphazard. So this can lead to a great discussion of the importance of adaptations and the importance of being able to adapt and change over time according to your environment. And so hope you like it. Good luck with it, and take care.